Hi, my name is Baudouin. I'm a PhD student from the lab of Sherif Abouelala and Michel Scott in the University of Sherbrooke, Canada. So you may have heard yesterday talks about the CD snoRNAs, and here I am here to convince you that HAC snoRNAs are also cool and in the context of pseudouridine level in the human ribosome. So I'd like to come back to the central point that the ribosomal is in the different branches of the living. And indeed, this centrality is reflected by the composition of the ribosome through these different species. So you will still find ribosomal proteins, ribosomal RNAs organized around a large and a smaller subunits. And even though there's a phenomenon of accretion where you can see the elongation and the increase in the number of protein, you have still this centrality in this composition. But with this accretion phenomenon, also a space was created for heterogeneity through evolution. And this heterogeneity, it was something we have evidences accumulated about the ribosomal heterogeneity since over a decade now. And so this variability in the components of the ribosome can be expressed through different layers. One first layer could be the proteins and the parallel proteins, notably. Also the post-translational modification that can appear on the proteins, and their abundance, because some of the proteins may not be necessary for the ribosome to keep performing, to maintain its integrity. A second layer of heterogeneity would be the ribosomal RNA variants, so this is really this skeleton where the different protein and other factors are organized around. But sometimes you have variation in the sequence, mostly in expansion segments, and so that's also bringing heterogeneity. One last layer is the layer of post-transcriptional modification happening on the ribosomal RNA. In human, it's mostly 2'-methylation and pseudouridylation, and here I am really interested in the latter. So pseudouridylation is an isomerization reaction where you take a uridine, you turn it into a pseudouridine, you induce a rotation of a nitrogen, creating the opportunity for a new hydrogen bond. And so this is recognized as a meshing, as a stabilizing modification, kind of modification in the ribosome. And so this modification, it is guided by a specific class of small non-coding RNAs called the snoRNAs, for small nuclear RNAs. And this is the HAC family that is guiding the modification by base complementarity on the ribosomal RNA. So HAC are called after those two motif boxes, A and H and ACA. And HAC snoRNA, they are really structured. They host two pseudo-relation pockets, as you can see, one here and one here, but they are just guiding the modification. They actually need 4 other core binding proteins to form a snoRNP complex that will be able to perform the pseudo-relation. And I'd like to point out the major role that the discarin protein is taking, because it is the protein with the catalytic activity for to perform the modification. And so in the human ribosome, there are about 108 pseudouridine sites, as you can see. So a bit more than 60 in the larger subunit and 40 in the small subunit. But there are not, the pseudouridine, they are not treated equally, we could say, because there is a real dichotomy between pseudourylation sites that will be stable, meaning that they are modified in almost every ribosome, while other sites can be variable. They can show variability, a frequency of modification that is not even, and that can depend on different contexts. And this is actually key to understand the importance of pseudouridine and of post-transcriptional modification in the ribosome. Because pseudouridine is also a very widespread modification on different type of RNAs, coding and non-coding. It is involved also in many different processes, such as RNA splicing or translation. And it has been shown in several studies that the dysregulation of the pseudouridine level can lead to the development of several diseases, including cancer. Unfortunately, there's a big gap between the modification being dysregulated and all the impact after on the translation program coming by the modification in the structure of the ribosome or other factors that can be included here. So here I'm showing you kind of a review of the different modification in the ribosome. If you count the pseudouridine on this figure, you only have 3. And so we have very little information, and there's a real need to understand the bigger picture, like here. Understand the real dynamics that can really play a role and lead to the dysregulation and the disruption of the translation programs. And so this is kind of the ambitious question I am asking here. So I am trying to understand more about the determinants that are driving this heterogeneity at the pseudouridylation level. And so to try to answer that question the best I can, or at least to shed a bit of light on the problem, I took a broad repertoire of healthy cells to quantify on one hand the snoRNAs and on the other hand the pseudouridylation. So to do this, we took different healthy tissues coming from sexual and non-sexual organs, coming from men, women, and on top of that we also used stem cells to try to simulate a different context where the system could be really different. So to quantify the HACA snoRNA expression, we use a classic RNA-seq, except we use a thermostable enzyme coming from a bacteria that is so working at higher temperature, something that is really useful for RNAs such as HACA snoRNA that are highly structured. To quantify the pseudouridylation level, we adopted a coupled approach using hydropsysic method, that is a chemically derived method that can quantify the pseudouridine score through a psi score. And so this quantification is made by is done after a cleavage of the uridine. So there is only the pseudouridine that remains here. And on the other hand, we also used nanopore sequencing, so taking the current intensity disruption as a signal to detect and quantify the pseudouridine in our different molecules. So here I'm kind of showing you the basic results where so you have a matrix of expressed RNAs. So here you have all the expressed RNAs along my different samples. And so if you group them if you group them, you will notice that there is basically one group of highly expressed RNA that are constantly expressed along all my samples and my organs. You have a second group of variably expressed RNA, so it can be context dependent, and a third group where there are a lower expression in general. And this expression also has an interesting tendency to, because you can see that snoRNA might be more expressed in sexual tissue compared to non-sexual tissue. So now, if we look at the other part of the picture, at the pseudouridylation site. So here I am showing you the different pseudouridylation site in my different sample. It is average, so you have the average psi score here. So the closer to 1, the more pseudourylated it is in my sample. And here you have the variation of those different sites. And one thing that is striking, it is the amount of sites that show a complete or almost complete and constant level of modification. So this is really a stable population, and where you can see at least 2/3 of your site being really in this specific area in this graph. And so this is kind of a discrepancy because I showed you a variable snoRNA that is supposed to target pseudouridine landscape, and now I'm showing you a static pseudouridine landscape. And actually, it is a bit worse than that, because if you connect the snoRNAs that are supposedly guiding the modification, and you look at them, here is what you got. So, for instance, I'm showing you here the group 3 of snoRNAs, which is supposed to be the snoRNA, the group where the snoRNA are the most expressed. And as you can see, they are actually modifying a small portion of the complete and constant site. So there is something, the connection is missing here. And so if you look more into details and really try to correlate the expression of the HAC snoRNA with the level of modification, here's what you can see. So we did the Pearson's correlation. And so if you consider that a good correlation is above 0.4, you will find actually about 20 strong sites that associate strongly with their snoRNAs. So those associations can look pretty decent actually, with a nice logarithmic dynamics. But for most of the sites, you have an unclear association that is remaining. Many explanations are possible. One of them I'm showing here is the fact that snoRNAs, they come into copies. And so sometimes it's really hard to say which copy has an effect, or if it's a synergic effect or alternative effect. So that's one reason that could explain that. So then we look to explain this and explore a little more the pseudouridine dynamic beyond the simple and the sole expression of HAC snoRNAs. So we try to take into account different parameters, taking into account accessibility, for example, collecting data about protein fixation, GC richness, proximal modification on the sites, also the availability of the snoRNAs, the way it is encrypted in the genome, the different way it binds to the core proteins, and also another layer with the conservation of the site, if it appears to be specifically human, specifically mammal, etc., etc. And so by looking at all this data, we stumble across something interesting about the interaction between the snoRNAs and the discrete protein. So indeed, if you took if you look at the snoRNA expression after a discrete knockdown, you will see that my cluster of highly expressed snoRNA is less sensitive proportionally to the discrete knockdown compared to the two other clusters. So we went to explore and investigate this, and so we did a time course discrete depletion to look at the specific effect of a discrete scarcity and how it could impact also the expression, the individual expression of HAC snoRNA over time. So we went with that. We did target sequencing, and we managed to isolate a few time points that then were clustered and showed us actually different dynamics between the snoRNA and the discrete, revealing three type of dynamics that seems to be pretty clear and discriminate clearly different behavior between the snoRNA and their pseudo affinity they could have with the discrete at this level. And so today I try to show you a model we are building. So this model is using different methods to try to make sense between the snoRNA and the different pseudo-relation level in the cell. As we have seen, there are most of the sites do not really associate with the expression of the HAC snoRNA. So we only have about 21 sites that show a good correlation with the expression of the snoRNA. So what we did is we went to disrupt the system. We went to lower the level of discrete to simulate some kind of competition between the snoRNA, trying to see if they could have a pseudo affinity at the individual level that could explain the fact that we don't the level of modification could be like more regulated through the snoRNP and not directly the snoRNA. And we actually found 3 different clusters that seems to show meaningful different dynamics between the snoRNA and the discrete. And so with that, I'd like to thank my lab for its support, my different collaborators, and you for your attention.
Thank you. Any questions? Maybe you can say something about when, during the cell's life or in differentiation, does the dynamic of snoRNA important?
Yeah, so the snoRNA, they are canonically recognized as modifying ribosomal RNA really early in the biogenesis process. So this is key to the biogenesis process. So the modification is really early. You cannot really export the different subunits if you don't have this modification. That's also why some claims are that this modification are like quality stamp that need to validate the ribosomal RNA transcript before further maturation. Yeah.
Okay, thank you. So if there are no further questions, we're going to move on. Thank you so much.
Thank you.